Thank you so much for that introduction. And thank you so much for the opportunity to present in this symposium and to speak with you about my research on self-regulation. I'm deeply appreciative of this opportunity. I want to present a model of self-regulation and consider some of its implications for development, and then sort of interrogate that model with data from a longitudinal sample that Nancy mentioned that my collaborators and I have been conducting, and with results from an education experiment as well. So with that, now for anyone with more than a passing familiarity with research on self-regulation in children and adults, you know that we're confronted with a variety of terms, constructs, definitions that seem to define this phenomenon of self-regulation. And it's natural to wonder, to what extent are these really terms for the same thing? Are they synonymous, or are they terms for different things, or are they kind of overlapping or not really overlapping very much? And to tell you the truth, I have no idea, although I'm an expert in this area. But I do think it makes sense to begin to group these in particular domains, to think about the more cognitive aspects of self-regulation, to think about the more emotional, reward sensitivity-based aspects of self-regulation, to think more about the personality, temperament manifestations of self-regulation, and to think about the ways in which these different domains work together in a system of self-regulation. A system that's characterized by recursive processes, that's both top-down and bottom-up. That is, whereas we know we can use executive functions to regulate attention, to regulate emotion, to regulate physiologically, by that same token, we know that as experiences registered physiologically, emotionally, and more automatic, less volitional processes, that's going to have implications for our ability to control our attention when needed and to engage the executive functions. It could facilitate it, but it can also really undercut it, which is more familiar to us as the stress phenomenon. I think there's some advantages to thinking about self-regulation in this way. One is it allows for sort of ready incorporation of a genetic level of analysis. We know that we differ in a bottom-up perspective in variants of genes that are associated with sensitivity to catecholamines and glucocorticoids that are expressed in the stress response. And as well, it incorporates a sort of an epigenetic process, both top-down and bottom-up, with the emphasis on circulating levels of glucocorticoids and the ways in which these levels may influence processes of gene expression and epigenesis. As well, I think it's valuable to think about this model because it's well grounded in a tradition in cybernetic theory going back 60 or more years, in which we think of these biological systems as a system that is self-regulating, that it's going to be adaptive in response to the context in which it's situated. And then finally, I really like this model or thinking about self-regulation because it has kind of an amazing neurobiology behind it. We know that as experience is registered rapidly, I'm trying to find the cursor. Oh, here it is. Thank you. As experience is registered rapidly in midbrain and limbic structures and the production of catecholamines and glucocorticoids, they feed forward. Whoops. I got to go back. Rats. Sorry, guys. I got to be careful with this mouse. So here, let me use this one. As they feed forward to prefrontal cortex, they are potentiating synaptic activity in prefrontal cortex and feeding back on this system, this loop, to sort of optimize levels of arousal and attention, as if it's, we're saying, hey, stimulation's occurring, pay attention, be goal-directed. And the ideas behind this system is that the catecholamine levels actually function literally in an inverted U-shaped fashion, potentiating activity, neural activity in prefrontal cortex associated with executive functions, and as measured behaviorally by working memory tasks, by inhibitory control tasks, by attention shifting tasks. But as catecholamines and glucocorticoids rise beyond an intermediate level, they begin to shut down activity in prefrontal cortex, in the seat of executive functions and the volitional control of attention. And as they rise further and further, indicating that the organism is under stress and a little freaked out, we see the sort of loss of this top-down prefrontal control and an increase in more reactive, less volitional responses to stimulation. And of course, this research is the research of Amy Arnston at Yale University, and well established through pharmacology and through single unit physiology in non-human primates. But it's very helpful to me in sort of providing a basis for understanding self-regulation as a system, and then beginning to consider some of the implications for development. If self-regulation is working in this way, how would we begin to study the development of higher order executive function abilities in early childhood, and the volitional regulation of behavior and the volitional regulation of attention? Well, we would focus on reactivity and regulation of the lower level systems early in development. They're in advance of, presumably, development in the higher order systems. And we would look there for the extent to which activity there is predictive of later executive function development. As well, it really helps sharpen our focus on experience in early childhood in terms of individual differences in the sort of parenting that children receive, as well as their early care experiences outside of the home, with the idea that the early care experiences are shaping this self-regulation system. They're influencing the development of this neural network that's important for the regulation of behavior and brain development. And through that are shaping sort of a more reactive versus a more reflective phenotype. That's the theory behind this model. And the idea, it's a theory based in a psychobiological principle referred to as experiential canalization, forwarded by Gilbert Gottlieb, one of my mentors and spiritual advisors, on the way in which environment and biology combine to shape development in ways that are appropriate for the context in which development's occurring, right? So this canalization process is a characteristic of biological, psychobiological systems. And my colleague Sibel Raver at NYU and I have really thought about the extent to which this characterizes self-regulation development in children, this appropriate development within context and highly disadvantaged context, a more reactive phenotype, and highly supportive context, a more advantageous phenotype in terms of reflection. But really, the questions have to do with the extent of developmental malleability in this system. To what extent, when children are moving from an advantageous to a disadvantageous context, or from a disadvantageous to a more hopeful advantageous context, are we seeing changes in self-regulation in the systems where we would expect them? So we've been interrogating this model with longitudinal data from the sample that Nancy mentioned, the Family Life Project. This is a wonderful study. It's a longitudinal population-based sample of children and families followed from birth really in and around small towns in rural Pennsylvania and rural North Carolina. It's a joint project with my colleagues at the University of North Carolina at Chapel Hill and Penn State University. In its first 2 rounds, it was funded as a program project by NICHD, and the projects were focused on family ecology, parenting, work-family relations, language development, and of course, self-regulation. And it has data collection in the home at these time points with follow-up through the 2nd grade. And I'm happy to say we recently just got funded to follow the sample into early adolescence, finally. And I'm also happy to say that the first phase of the data collection are now publicly available up through 36 months. So if you're interested in this data set and in longitudinal analysis, by all means go to the ICPSR with this accession number and download the data sets. And you can even check some of the stuff that I'm going to talk to you about today, if you're interested in sort of checking my work. So what I'm going to focus on is really parenting, and parenting sensitivity is the thing that we're most interested in here in the Family Life Project. So we're collecting that in a structured free play interaction. We're collecting saliva from children in infancy and the toddler periods. We're collecting that saliva both at baseline, adjusted for the time of day, and looking at the stress hormone, the glucocorticoid hormone cortisol, as an indicator of activity in the physiological system. And we're also collecting it in response to a motion challenge, so a toy removal task and a scary mask presentation. And we're looking at relations of parenting and child physiology, both at baseline and in response to the task, and emotional reactivity and emotional regulation that are coded second by second in response to these tasks. And we're relating those to measures of executive function with these types of tasks that we developed for longitudinal use. And so just to recap really quickly, we're getting cortisol from saliva at these time points at baseline and in response to emotion tasks. We're looking at parenting observed in a structured free play, measures of risk in the home, risk in the household, and then executive function as children enter preschool, right? And here, just a first analysis looking at just the basal samples of cortisol collected, assayed from saliva samples collected at baseline. And what we're seeing here is an expected or typical decline. I don't think we really had data like this before from infancy up through the toddler period. For children in the dark line in more stable, less chaotic homes, but here for children in more chaotic, less stable homes, we're not seeing that decline. It's as if the cortisol levels are elevated and staying elevated, and thereby being less flexible in response to experience. The analogy would be sort of like a home in which the thermostat's not working well, but there's going to be unpredictable severe cold out. So you might just turn the heat on and leave it on. The house may burn down or become damaged in some other way, but it will protect against that unpredictable severe cold. However, in a home in which the thermostat's working well, it will be flexibly regulated in response to the outside temperature, right? So that flexible regulation, we think, is really being driven by maternal stimulation and maternal sensitivity. So we then looked at the cortisol in response to the emotion challenge, and what we found is at 7 months of age, the children experiencing more sensitive care as measured by that free play interaction were showing an expected increase and decrease in response to the emotion challenge. Here's the idea is that the physiology and the behavior are going along. The children in the dashed line experiencing the less sensitive care in the more chaotic homes, they're just as emotionally aroused by the tasks, but the physiology and the behavior are less well coordinated. So the idea here is that maternal behavior, early caregiving behavior, is organizing the systems early on to work together rather than individually. And then we follow the sample forward to 15 months of age and follow the sample forward to 24 months of age and see similar sorts of patterns. In particular, at 24 months of age, this is the third time that children have seen these tasks, and some of them are still emotionally aroused by them and some are not. And what we see is high levels of maternal sensitivity are associated with high levels of cortisol when children are exhibiting an emotional response. However, when they're not, cortisol levels are low overall. So again, this idea of a coordination of the physiology and the behavior in the context of high quality care. And then, of course, we have this measured longitudinally so we can begin to ask interesting questions about change in maternal sensitivity because it is changeable and can change. Looking at this change in behavior and looking at change in cortisol was what my, at the time, postdoc Dan Barry decided to do. Dan's now an assistant professor at the University of Illinois, but he had this brilliant idea. If we see within-person changes in maternal sensitivity, within mom changes in maternal sensitivity, will we see within kid changes in cortisol? And of course we do. And it's this dark bar here. And the really fascinating thing about this analysis is it's in the lower levels of sensitive parenting that we see the big effect of change. That is, moms who are initially low in their sensitivity and increase their sensitivity over time are having a big effect on children's cortisol levels, which is good news, right, in many ways, and exactly what we would want to see. And of course, important because what we see is that the positive, this more sensitive parenting and cortisol levels are predicting executive function at age 3, the first time point when we're able to measure executive function, right? It's mediating some of the risk characteristics in the home through parenting, through cortisol, to executive function in support of this model of self-regulation and self-regulation development. So of course, I've been talking about sort of baseline low levels of cortisol, but also the flexible regulation of cortisol. So which is it, Clancy? You can't have it both ways. Well, you kind of can. So we then, based on that, we hypothesized that moderate variability in cortisol levels in early childhood around a low average mean would be associated with the highest levels of executive function at school entry, if you follow me on that. And that's kind of exactly what we found there with this. So I think there are several implications of thinking about self-regulation in this way and fostering child development, particularly for families in poverty, and the idea that maternal sensitivity parenting behavior is changeable. As part of the work in my lab and shared with Sibel Raver and our labs jointly, we're part of a group that is a consortium of 6 universities around a buffering toxic stress consortium. These are funded by the Administration for Children and Families, who oversees the Early Head Start program. And our goals in this really are kind of validation of the relations among parenting and poverty, stress physiology, and child outcomes, but also mom outcomes, mom self-regulation, and looking at influences of mother self-regulation on child self-regulation, a mechanism of intergenerational transmission, looking at the potential for implementing a high-quality parenting program through the Early Head Start home visit, and conducting an experimental evaluation of that. So simple tasks like that. We're getting through this study now, and we'll have data, I think, soon from the consortium on the viability of doing these things and the potential for high-quality parenting support as a focus for the types of services that are being provided by ACF. And then I think there are also some important implications for education I just want to wrap up with. This thinking about self-regulation in this way as a psychobiological model, it's one manifestation of this sometime uneasy relationship between neuroscience and education. The idea that what we know about the neurobiology of the brain and brain development can really inform educational practice, can help us answer the question of what we should do and how we should do it and what we should measure. And I kind of think of this as an early education version of working memory training, if you're familiar with the working memory training literature. That literature, Torkel Klingberg, Susan Yagi, have shown that the brain is plastic even into adulthood, right? When young adults do repetitive practice on working memory tasks, they get better at the tasks, and we see concomitant changes in the neural networks that underlie this behavior. The problem is, very little of that generalizes to anything that we're really interested in. If you want to get better at working memory tasks, you can do it. So in education, we can build on this, but let's embed the tasks, the working memory tasks, in things that we want children to do, where we want them to generalize. So what would that look like in terms of education? Well, it would look like reflection on experience, planning, self-direction, even in 3, 4, 5-year-olds. And that would be a great way to try to embed self-regulation in children's development and their early educational experience. So rather than have to invent the wheel, I found a program, there are probably several programs that do this, but one that really makes this the explicit focus of its pedagogy is Tools of the Mind. And there's a pre-kindergarten version of Tools, and there's a kindergarten version of Tools. And I don't have time to go into it because I'm just about out of time. But there's lots of information on Tools of the Mind on the web if you're interested in this. But the bottom line is we conducted a randomized controlled trial of the kindergarten version with this many school districts, that many schools, that many classrooms, and that many kids. And importantly, schools range from 3%, very advantaged schools, to very disadvantaged schools in terms of free and reduced lunch. We had measures of executive function, control of attention, speed of information processing, academic outcomes, and of course, cortisol. And what we really found in the sample overall is not much to write home about. There were small effects. So you can think of a standard deviation as about a year of growth. So in the sample overall, we saw small effects on working memory, executive function, reaction time, a little bit on math, and a little bit on reading. But when we looked in the high poverty school, that's when we really got the eye-popping results, right? And there we saw pretty big, about on average, a half standard deviation effect in these high poverty schools for this very focused program on self-regulation development. So looking at across these outcomes, including reasoning and vocabulary. So a very interesting and provocative set of findings. I think they're really relatively few evaluations like this. This educational research is hard to do, but we were able to follow the sample forward into the first grade and found effects on growth in reading and growth in vocabulary. So with that, I have to stop. I'm out of time. I would love to talk with you more. Take-home message from this talk is let's, as much as possible, make our scientific understanding of self-regulation and the neurobiology of self-regulation inform what we do, how we do it, and what we're measuring in terms of the outcome. So with that, I want to thank all of my collaborators at Penn State, UNC Chapel Hill, and New York University, the folks with Tools of the Mind, and appreciate the very generous funding from NICHD, the Institute of Education Sciences, and the Administration for Children and Families. And thank you for your time and attention.